You've probably heard about antibiotic resistance, these superbugs that don't seem to respond to our usual medications. Well, resistance doesn't just come from nowhere. There are several mechanisms of genetic variation in the world of bacteria. However, any new change in genetic code comes from a process called mutation. Mutations happen when bacteria make mistakes copying their DNA. So, today, let's talk about genome replication in bacteria and see how this process is both elegant and also can lead to stuff like antibiotic resistance. Bacteria usually have a single small circular chromosome, and many also carry plasmids. Plasmids are like bonus DNA. These small extra genetic elements can carry useful traits like antibiotic resistance genes. Both the chromosome and plasmids need to be copied or replicated when a bacterium divides. Replication starts at a special spot called the origin of replication on the chromosome. Imagine a zipper being pulled apart. Helicase is the enzyme that unzips the DNA, breaking hydrogen bonds between the strands. But unzipping creates supercoiling ahead of the fork because the chromosome is circular, kind of like a twisted telephone cord. Increasing tension is created in the chromosome as helicase continues to pull the strands of DNA apart. That's where DNA gyrase steps in, relieving the tension by breaking and reattaching DNA so the strands don't snap from tension. Now that the strands are open, we need to start building. But here's a problem. DNA polymerases can't just start from scratch. They can only extend an existing chain, and they require a 3' hydroxyl to operate. So, primase is an enzyme that makes a short stretch of RNA called a primer. Why RNA? Because RNA nucleotides can be built without a primer. Once the primer is in place, DNA polymerase 3 gets to work, adding nucleotides in the 5' to 3' direction. Remember that DNA is always built in the 5' to 3' direction, which also means that the template strand is read in the opposite direction, 3' to 5'. Here's where things can get tricky. DNA is anti-parallel, so only one new strand, the leading strand, can be built continuously, following the direction of helicase. The other strand, the lagging strand, has to be made in fragments. These are called Okazaki fragments. After a stretch of DNA is made, we need to backtrack by creating a new RNA primer closer to helicase at the replication fork. As helicase continues in the opposite direction of the lagging strand synthesis, the process will be repeated and the lagging strand is built in fragments. Eventually, the RNA fragments need to be cleaned up. We don't want pieces of RNA in our DNA. So DNA polymerase 1 will come through, remove the RNA fragments, and replace them with DNA. But we still have nicks between fragments in our DNA. That's where ligase comes in and glues all the pieces together. In bacteria, replication keeps going around the circle until the two forks meet at the terminus. Now we've got two full circles of DNA, but sometimes they're tangled like two linked keychains. These are called catenanes. Specialized enzymes cut and rejoin the DNA to untangle them. Or sometimes we have a scenario where the chromosome copies are fused at the end of replication. That's called a dimerized chromosome. Bacteria solve this with recombination enzymes that split the fused DNA into two proper circles, so each daughter cell gets only one copy of DNA. And here's the connection to antibiotic resistance. Every time bacteria copy their DNA, there's a chance for an error to occur. A random mutation in a gene might change a protein in a way that makes an antibiotic less effective. And if that mutation happens on a plasmid, it can spread quickly between bacteria. That's why understanding DNA replication isn't just textbook knowledge, it's a key to tackling serious problems like superbugs. So bacterial DNA replication is a team effort. We have enzymes like helicase, DNA gyrase, polymerases, primase, and ligase all working together to copy life's instructions. It's precise but not perfect, and those small mistakes are part of why we're in a constant arms race with bacteria.